This is episode 302 of the Stem Cell Podcast, Kidney Organoids with Dr. Zhongwei Li. Hello, everybody. This is Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Zhongwei Li, who's from the University of Southern California. He's on the podcast to talk about his research on kidney organogenesis, disease modeling, and regeneration. We've also got our usual roundup of recent highlights in stem cell news that's coming right up.
But first, STEMCELL Technologies would like to introduce their one-stop resource for researchers who are using or hoping to use organoids in their experiments. STEMCELL's Organoid Information Hub provides scientists with instructional videos, educational webinars, expert interviews, technical tips, and curated publications to help researchers set up and optimize organoids as a research model in their labs. Learn more about organoid culture from the experts at STEMCELL. Visit www.stemcell.com/discover-organoids.
Organoids! Oh boy, seems like ancient history that we were impressed by organoids. The last couple of years now, we've been talking mostly about embryo models. It's become ubiquitous. We're past the shock. The ethical conundrums may still be lingering, but the capability has advanced, I would say, incrementally over these last 2 years in really marvelous increments. Every one of these stories is a spectacle just because we're getting closer and closer to being able to recapitulate the miracle of self-organization and organogenesis toward practical as well as just, investigatory ends. I think this story, and you got a story right after that's similar, Arun, but I think this story is really a great leap forward, a big increment, in my view. And that's because I think it's really lowered the bar in terms of practicality and accessibility to the point where I think these models can now be applied in any lab that is able to work with tissue culture. All right, so let's get to it. This is a story in Cell from Jose Silva Lab. Silva Lab was transplanted from Cambridge to Guangzhou National Laboratory in China about 3 years ago. And over the course of those 3 years, they've been cranking away toward perfecting these embryo models. And it's really all based on the pieces of these models, which is the three blastocyst fates, primitive endoderm, trophectoderm, and inner cell mass, right? And these cells have been mixed and demonstrated the ability to self-organize into these early embryo-like structures, starting with Nicolas Rivron with the blastoids and then extending beyond by myriad groups to make these gastro-like embryo models in mouse, human, many different species at this point. The current state of the art, although state of the art is a moving target, as you're going to underscore in your next story, Arun. But currently, the methods involve mixing, to say the least. You got to mix the cells. Most of the time, you're mixing embryonic stem cells with representative cell types of the extraembryonic endoderm, or these Zen or primitive endoderm-like cells, as well as trophectoderm stem cells or trophoblast stem cells. And you generate these cells sometimes by exogenous expression or enforced expression of like GATA6 or GATA4 in the case of the primitive endoderm, or CDX2 in the case of trophectoderm. Or you can differentiate these cells through, cell induction processes. As I said, the progress has been incremental, and we're closer to getting the transgene free. But there's still a lot of cell mixing And having to mix these cells sometimes with the transgenes introduces these variables that ultimately undermines the efficiency and reproducibility of these models, and also the fidelity to like the ground truth of the embryo in vivo. Um, let's turn to early blastomeres. We've talked about all the different cells, the 2C like, the naive. There's all a spectrum, a broad spectrum of potency within these pre-implantation embryo stages. But the blastomere is the one that generates the trophectoderm, uh, and the primitive endoderm and the epiblast, right? And so these are like a founder cell. And Silva Lab, initially, when they were still at Cambridge, they observed that you could increase STAT3 activation, and get a developmental stage that was a bit earlier than the induced pluripotent stem cell. And then later, one of the first things they did, or the first big stories that they had after moving to Guangzhou, was generation of these morula-like cells that were able to contribute to all the different cell types in the blasts, notably intercell mass and extraembryonic. Um, and what was interesting is when you looked at these cells, they had co-expression of OCT4 and NANOG, as well as GATA6, which are like seemingly antagonistic, representing these two fates. So that was the approach that they used here, uh, scoring on the co-expression of those two factors. They developed this chemical reprogramming screening approach to generate these founder like cells. With You should look at the paper. This is such a huge amount of work, reminiscent of Honky Deng's mastery of the chemical libraries. And what they ultimately converged on here was a recipe to induce mouse, in this case mouse, embryonic stem cells to these 8 to 16 cell like embryo founder cells that correlate with the blastomeres at that stage. And they showed with embryo complementation in vitro, in vivo, that they can specify the embryonic and extraembryonic lineages in the blastocyst. Uh, then they could make embryo models, and this is the key. They could put in just these founder cells into these agrawells, microwells, and without any cell mixing, and generate embryo models, which with very high efficiency that would go all the way, not all the way, but would advance through organogenesis. And this was really the most amazing thing about this paper, is the visuals and the scale. They would show wells that were just filled with embryos that I don't want to say they were indistinguishable from the ground truth, but they looked a lot like it. Arun, you got to look at these cardiomyocyte, looping hearts beating on time over the course of minutes. They showed somi pairs, fore, mid, hindbrain, sorry. Uh, Looping heart tube, as I said, optic buds, allantois, tail bud, primordial germ cells, well-defined gut. So really pretty advanced organogenesis stages. And as I said, the scale of it, the efficiency of it, and the accessibility. Once you get these cells, you just pop them in, don't have to mix, no transgene expression, and you're getting these beautiful mirrors, although imperfect still, but really close and scalable recapitulations of, in this case, mouse embryogenesis out to like E 8.5, 9.5. So really amazing work here, Arun. I really think a watershed move forward for the field and making these models accessible to all labs.
That right there is the key point, is the accessibility, because the two major things that a model like this, and even the paper I'm about to talk, are emphasizing, is that transgene free and no mixing of the cells. And that, like you said, really lowers the bar in terms of anybody who has access to pluripotent stem cell culture being able to create some of these models in high throughput. And of course, I for sure, the first thing I saw or went to in the paper was the videos of the contracting heart tube and the beating hearts. They had like, 7, 8 different embryo models lined up, and you could see their heart tubes actually contracting. Really, really impressive stuff. And of course, they did the deep characterization in addition to the visualization. But I totally agree with you. It's the accessibility of this. And I'm going to talk about the another way this is becoming more accessible through Jacob Hanna's work. Of course, for them, they have more of like this custom roller culture approach, which it's more of a technical lift to be able to implement that in the lab. But I think the bar here is pretty low for anybody adapting this. I mean, it's still not perfect, and they've gone ahead and said that. And I think one thing they specifically mentioned that their embryo models here lacked are the a proper orientation of the outer mural trophectoderm that the true embryo actually uses to attach to the uterus. So it looks like implantation here is still not it's not ready for prime time when it comes to implantation. And there's some differences when it came to the mechanical stiffness and mechanotransduction, all that kind of stuff. But I mean, just the visuals here are extremely striking. And I'm getting to the point, and I actually alluded to this in a previous episode, where I might start thinking about bringing embryo models into my own laboratory because it's just becoming so easy to do, right?
Yeah, and the morphogenesis that you got, you zoom in on those hearts and they're looped up nice. But also, yeah, the as you're saying there, the fundamental question I'm left with is like, how long before we put these in a uterus and they form an embryo, right? And I think that that's another interesting takeaway here, is that it seemed like there's some fundamental, I don't want to call it deficit, but there's differences for sure in the orientation of these things that may be what has undermined the formation of like proper embryos in vivo, or implantation. But like honestly, part of the beauty of these models is that you don't need to implant, and you can still get all these structures and cell types that are broad ranging. I mean, we're talking about hematopoietic stem progenitor, primordial germ cells, some high target cell types. So really amazing. Interested to see how long it's going to take for this to dribble down to human.
Yeah, and if we're interested in unlocking that black box of early development, I think these things are, you know, definitely ready for prime time. It's later that certainly there needs to be more work done, implantation, all that kind of stuff. But I mean, the progress in this field has been astounding in the last 5 years, even the last 2 years. So who knows what the next couple years are going to bring? And I think the next paper is a perfect complement to the study that you just presented. This is a cell stem cell paper from Jacob Hanna's group, but there's a lot of very high profile folks on this particular paper, including Konrad Hosodinger, number of other folks. First author here is Alperin Yilmaz. Yeah, the title here is alluding to something that we were just talking about, and that's the importance of transgene free approaches in developing these embryo models. So that's literally the first word here in the title of the cell stem cell paper: transgene free generation of mouse post gastrulation whole embryo models solely from naive embryonic stem cells and iPSCs. And again, also, same thing, no mixing, right? So we're talking about gastrulation, early embryo models, um, pretty much exclusively from these naive embryonic stem cells, which is really critical to drive this forward. These things can give rise to the embryonic lineages, but also extra embryonic lineages have been produced as well. And like you alluded to in your previous study, the existing protocols usually rely on the separate induction of extra embryonic lineages and then like overexpression with transcription factors to actually induce differentiation in trophectoderm, primitive endoderm. But here they're able to show using mouse naive embryonic stem cells combined with naive induced pluripotent stem cells that they can be simultaneously co-induced through some pretty extensive signaling pathway modulation to generate these primitive endoderm and trophectoderm extraembryonic cells that can self-organize into like E8.5, E7.8.75, importantly, transgene free embryo models. So um, it's a technical heavy lift from the Jacob Hanna group, where they developed kind of multiple approaches to get to this end goal. They actually had this like alternative condition naive media that also stabilizes these trophectoderm synthetic embryo competent OCT4 nanog naive ESC colonies that they're expressing all the things that you would expect, like antagonistic CDX2 and or the GATA6 extra embryonic fate, different regulators, but they're still poised for the trophectoderm and primitive endoderm differentiation. So take a look at this one. It's basically a bunch of different inhibitors, small molecules, just like what you're alluding to, really small molecule driven here. No transgene overexpression, a bunch of HDAC inhibitors, GSK beta inhibitors, TGF inhibitors, um, to force the production and early differentiation of these cells. Um, no mixing. You got this Agarwell approach, which also Nicolas Ruron is using as well to really kind of force these things together. And then one of the technical innovations that Jacob Hanna's group has created to make these things progress pretty far along are these roller cultures. So they incorporated the roller cultures here. So they had this 3 days and the electronically controlled ex utero culture is what they call it. And then, ultimately, of course, the characterization of these transgene free embryo models. Again, they're able to grow until post gastrulation, early organogenesis, all the kind of the same stuff that you showed in your paper. They're able to see here. So it's I think this is a really nice complement to the study that you showed. I think slightly different because they're using the roller culture here, which is, as far as I know, still a technically challenging approach and is something still pretty much exclusive to Jacob Hanna's group. But yeah, you got the transgene free approach, you got the no mixing approach, you got the small molecules that are driving all of this. So, you know, really great story here.
Yes, and I don't want to say that the, the Silva story is simple, but this story is so complex, which is not to say inaccessible. I think it's just reflective of how long and how, the Hannah Lab has been in this and how deep they've delved into developing the tool set here. But, for me, the greatest takeaway, and it's no coincidence these came out a day apart in Cell and Cell Stem Cell, because it's a super competitive field and these stories are really complementary. I want to say maybe some overlap, but in a good way here, I think, showing that, these beautiful representations of, mouse, in this case, embryogenesis, highly faithful, are being achieved in multiple labs. So this is what's so great about science at its best, is the reproducibility across groups. And I can't wait to see all of the collaborations that stem from this development and the Silva development. I'm sure these guys are all working together at this point.
Yeah, absolutely. I mean, these folks, the Hannah Group was pretty straight up about some of the limitations here. I mean, yeah, we talk about if you lay the real deal right next to the model, they're very similar morphologically and transcriptionally and all that kind of stuff. But there were some even visual differences, especially when it came to the cardiac tissue. So they had like a cardiac hypertrophy phenotype. These embryo models did. They were an very enlarged heart in comparison to the wild type or to the real deal. So they don't honestly they said they don't know why these things have bigger hearts than true embryos do, and obviously that's a very important part of development is proper cardiogenesis. So, um, but like what your paper was talking about, um, their hypothesis has to do with, mechanotransduction, and I think that's an element that still needs to be refined and tweaked in both of these studies and all of these embryo models, is because, um, yeah, that process is gonna be very different in vitro versus in vivo, so, maybe that's the key right there to taking even a further step down the road.
Yeah, well, I would also highlight the lack of any conduction, right? Any blood, any connection to maternal vasculature and or blood, a big part of the inductive events in embryogenesis and organogenesis have to do with the blood, that paracrine contribution. So I wouldn't be surprised if this may be something that is insurmountable given the divergence from like physiological development here from a mechanical standpoint as well as a cellular molecular standpoint. But nevertheless, I mean, at the end of the day, these are all models, right? We all know about what a model is. It's not perfect. It's wrong, but it's useful. And taking that pragmatic view, I think, this Schrepfer approach, hypoimmune approach, that's my segue, pragmatism. I love it. It's such there's a poetry to it. And for me, especially rooted in all the promises that were made back in the day when ES cells were first derived, it seemed like we were ignoring the fundamental limitation of what a heavy lift it was going to be to personalize these things. I mean, first, we were talking about doing somatic cell nuclear transfer per patient to generate personalized cells. Then iPS cells, we said, okay, problem solved. But still, uh, in the modern era, in the pharma ish paradigm, we need something off the shelf. And this hypoimmune thing is crazy. Last episode, you highlighted this NEJM story about the retention of beta islets, the hypoimmune beta islets in patients. Here, I've got a story that's in Cell Stem Cell, a clinical translational report that mirrors that advance, except here in the hematopoietic therapy world, specifically CAR T. And this is just sign of the times, but the Santa and Schrepfer are really way in the lead here, it seems. And we all know the concept, we all know the utility, increased uh, utility, like the economics of it, right? You can have something that's GMP, you can get from healthy donor cells, scale them, the cost and efficiencies are can't argue with. And it's all based on this idea of getting rid of that HLA, right? Right now we're confined by the HLA matching. And so even so, you got to do aggressive immunosuppression of these patients. So in the case of like, let's say, let's take that NEJM story where you had to take, allogeneic beta islets, say from a cadaver, you had to substitute one chronic therapy for another, in this case, immune suppression, which can have its own sequelae, increased rate of infections. So yeah, we can't argue with the hypoimmune concept. What's great about this is the success, right? So just brief review. The concept is based on depletion of the class 1, class 2 HLA epitopes, and then overexpression of this don't eat me signal CD47. And as I just mentioned, we have preclinical studies that were in mice, in primates for these islets, and then just a few weeks ago in human. Similarly, there's been this idea of using CAR T cells, CD19 CAR T cells that have been shown to work in humanized mice, have been shown to work in primates in the preclinical studies. And what's great about this in like the mice is that you put it into the circulating hypoimmune CAR T cells into the mouse, they get rid of the cancer. There's no rejection, they are retained. And then 3 months later, you introduce cancer again and they wipe that out as well. So there's this idea of like these sentinels that are engineered and introduced and then just surveilling to wipe stuff out, which you can imagine the applications not just for cancer but all kinds of degenerative disease, fibrosis. We've talked about it. Um, in this case, they had the first inpatient study, which was actually the aggregate of two investigations, this Ardent and Gleam in investigations, these two trials. But in aggregate, they amounted to 14 patients. And the endpoints here were really just to look at retention, their ability to evade an immune response. But of course, they also scored for the efficacy of these cells. And it was this primary T cell product. So this is not induced pluripotent stem cell derived CAR T, but with George Daly's advances, which he just talked about, I think last week, our last episode, that's soon to come. But in this case, primary T cells, they have the CD19 chimeric antigen receptor. They also have a knockout of this T cell receptor alpha, constant region, right? So that prevents the graft versus host disease. And then they have these hyperimmune edits, which are to get rid of the HLA, and they also overexpress CD47. So they had a mix of different combinations of these cells. Some had the CAR T, they all had the track, so they wouldn't cause graft versus host. I mean, these were human patients after all, but some had the CAR, some didn't. Some had the hypoimmune complement, some didn't. And they introduced these all into patients who were myeloablated, they were preconditioned, lymphodepleted. So I think that's an important note because one of the major applications of this is that you could precondition the niche in a way that didn't involve depletion, these like, chemotherapy essentially. In this case, use cyclophosphamide. But again, that's probably forthcoming. But in this case, they lymphodepleted and then treated these patients. Out of 14 patients, they had a range. Importantly, for most of them, there was no immune reaction, right? In two of the patients, there was they got in there, but they didn't deplete the CD19 positive cells. In three patients, they had a transient depletion of the CD19 cells. In six patients, they had a durable depletion of CD19. So that's the target. That's what you want. And rounding it out, they had three patients where it was the cells were rejected out of hand. And the important note, as they did in following up those patients, they found that, well, they surmised that in all likelihood, their conclusion that they drew was that those cells were rejected because of antibodies that were developed against the therapy. So they had previously been treated with therapies that had other components of these CAR T cells. And the judgment was that is what in these three patients was being reacted to and wiping out the hypoimmune cells. So it didn't have to do with the deficit in the approach. Fundamentally, even with those patients included, I think this is a remarkable success. And although it wasn't the primary endpoint, showing efficacy, these CAR T cells, I think, is very encouraging, showing that they not only evade the immune system, but they work. So this is first in human, I think, first report of many reports, not just in these patients, but in other patients who have been recruited. There's like hundreds of these adaptive immunotherapy trials going on already. So incorporation of this off-the-shelf component, which will increase efficacy and reduce, some of the negative side effects and sequelae of the treatment. It's new ball game. This is crazy. Buy stock in Santa if you can.
Yeah, really great stuff from Sonia Schreffer, who, like I mentioned, is now partly here at Cedar Sinai in my neck of the woods. And side note, we actually are. She's a co-I on a recent grant that we've gotten on something space related. So stay tuned for that. Hopefully, an announcement to come there. But she does a little bit of everything. I mean, it's not her primary focus. This is certainly her primary focus on the hypoimmune side of things. You know, this is also a kind of a reflection on the priorities of cell stem cell these days because it's almost 10 years ago, you wouldn't expect a paper like this to come out in Cell Stem Cell, but I know for a fact Chilchari, the editor in chief, is really emphasizing these short clinical reports, a reflection of the current translational nature of the stem cell field. And this is not by any means like a full scale clinical trial or anything. This is just looking at the immunology. It's like a brief immunological report of how these patients are doing, and you still have to look at the detailed downstream conclusions, downstream like safety, primary efficacy, outcome measures, and all that kind of stuff. But I think a very relevant paper for cell stem cell and kind of what they're emphasizing these days.
Yeah, I think it's exactly the right form because it's like a unlocking, right? That any jam was the right form for that. Maybe honestly, it was a little premature for that because he didn't have the long-term efficacy of the treatment in terms of the medical application. But I don't think it matters. These are such high impact and first of their kind studies that whatever form, as long as it's a major form like cell stem cell or NEJM, clinical or scientific. But here, I think it is perfect because it unlocks just the approach. It validates one of the more central elements of hypoimmune therapy, which now I'm sure. I mean, this isn't news, but is there anyone who isn't trying to go hypoimmune now? I'm sure we got to hedge on some level, but I can't see a future where the majority of cell therapies are not an allogeneic off-the-shelf hypoimmune product.
Well, Dalen, I'm not going hypoimmune, but that's because I'm not putting anything into people, and thank God for that. Not yet, anyways. But sticking in the realm of cell stem cell, this is not the cell stem cell podcast, but we do cover a lot of cell stem cell papers here, obviously. This is a paper coming from none other than Honghui Deng, who, of course, we interviewed at ISSCR in Hong Kong just a couple months ago. Building on, of course, the work that they've been doing on chemically induced pluripotent stem cells and so on. They're looking at, I'll just read the title of the paper: Reconstruction of Endocrine Subtype Complete Human Pluripotent Stem Cell Derived Islets with the Capacity for Hypoglycemia Protection in Vivo. And when we talk about all of these, stem cell derived islet stories, we very often focus on, you know, beta cells, right? Everybody's like beta cells, because those are the things that produce insulin, the beta cells. But the islet is comprised of a bunch of different cell types, not just the beta cells, right? So you got to, dissect this idea of the transplantation of PSC derived islets, which of course you got to have the functional beta cells that produce the insulin. And that's the source of the promising cell therapy for restoring glycemic control and diabetes. And we talked about that on that live episode. So check that one out if you haven't already. But if you want to recapitulate the complete endocrine composition in the PSC islets, you got to have all the different cell types, and it's still pretty challenging to do that perfectly, right? Um, so the other cell types, the other non-beta cells, have important metabolic roles, support roles in vitro and also in vivo. And here they're showing robust generation of non-beta cells in vitro. So they incorporated the non-beta cells and the beta cells to reconstruct the PSC islets, comprising, importantly, all five of the endocrine subtype cells: alpha, beta, gamma, epsilon. I'm missing something because I don't know my Greek letters. But yeah, there's five subtypes there, right? Ask the dang lab. I'm just a cardiac biologist. Sorry. So the all five subtypes of cells are comprising in these PSC islets, and of course, if you can make it in vitro, you got to apply it in vivo. That's what the Daily Lab and many others have been doing for years now, and Doug Melton certainly. So they applied it to the in vivo, right? They introduced these things, these fully reconstituted iPSC or PSC derived islets into a diabetic mouse model, showed what you would expect: reversal of hyperglycemia, robust protection against hypoglycemia as well, hyper and hypo. So, it's modulating insulin very well. Only 3% of the measurements actually falling below the critical 54 mgs per DL compared with 59% in the non-reconstructed controls. So they're doing a really good job in maintaining the proper glycemic levels. And then they suggested a restoration of previously defective counter-regulatory response in these reconstructed PSC islet recipients. So it's almost like a sequential treatment here, that these things are actually very effective in restoring proper insulin regulation, right? So it's kind of a nuts to like you're going from start to finish here. You're making the diff to actually make all 5 subtypes in vitro here, of that are comprising the PSC islet, not just the beta cells, but everything else in there. And then you're applying it. So you're establishing this strategy to actually control the relative abundance of the subtypes in those islets. So it's almost like calibration in a way. You got to calibrate the right numbers of the cells in those islets to get the best shot on goal for regulating, hypoglycemia, hyperglycemia, and providing that proper homeostasis for insulin regulation, right? Because that's what it's all about. It's about, we can make these things now and we can even put them in people, but how do you regulate glycemia? How do you regulate insulin secretion and production indefinitely so that in the future this is a truly an effective cure? Um, and I think this is a great step towards that.
Yeah, this I'd love the idea that, the we've arrived at the cure and now we're one-upping the cure with an optimized cure that is even closer to physiology. And I think it's a really important thing, not just for, as you said, it's a great analogy, best shot on goal, right? To calibrate the inputs. But I think as we start to think more about transplanting these structures, from an engineering standpoint, right? You want to understand and carefully control the balance of these cells in order to ensure their longevity, not just their function. And I think there's a lot of unknowns about the paracrine relationships among these cells. Of course, we focus on their functional contribution to the organ and homeostasis, right? Glycemic homeostasis. But I think there's a lot of biology there that's underappreciated. So why not build the thing how it's meant to be built, right? If we want it to function the way it's meant to be built, meant to function, let's get it right. So this is in keeping with the Deng Lab's general approach, which is focusing on the minutia to optimize the outcome.
Yeah, absolutely. And, we always talk about these like, magic bullets, single cell types that would be able to restore proper function in, say, the pancreas or even like in the brain when it comes to uh, dopaminergic neurons, right? It's we always think about single cell types that are doing the trick here, but of course, no cell truly functions in isolation, especially in something like an islet, right? Like you're alluding to, plenty of paracrine input from different cell types to get the true proper regulation of insulin in this situation. But you can think about how you might be able to apply this concept to other tissue types as well. Of like hepatocytes have certain massive paracrine signaling that occurs in the liver as well. And cardiac tissue is not just cardiomyocytes, but those fibroblasts, endothelial cells provide a lot of really important signaling. And similarly, glial cell types in the brain. And the brain is not just a bunch of neurons, you have a bunch of support cell types. So you got to think about those auxiliary cell types because they're doing important stuff.
Absolutely. We can't do it alone out there. Arun and the cells can't either. We're not going to be alone long. We got Zhongwei Li joining us in just a moment. But before we get to talking there, I got a quick message from STEMCELL Technologies. Take your human pluripotent stem cell cultures further with M-Teaser Plus from STEMCELL Technologies, the most widely published medium for feeder-free human ES and iPS cell maintenance, is now formulated for enhanced performance and versatility. M-Teaser Plus reduces medium acidosis for more stable cultures all weekend long. To learn more, visit www.stemcell.com/mteaserplus. All right, guys, today on the show from Arun's neck of the woods, we have with us Dr. Zhongwei Li, who is Associate Professor in the Department of Medicine at the Keck School of Medicine. They're in the Eli and Lilith Broad Center for Regenerative Medicine and Stem Cell Biology. Dr. Li and his team aim to bridge basic studies of kidney organogenesis with translational applications of kidney regeneration and disease modeling, with the long-term goal of rebuilding the kidney. Hey, no sweat, no pressure there. They achieve this by combining state-of-the-art stem cell technologies and engineering methodologies. Zhongwei, thank you so much for joining us on the show today.
Thank you for having me.
Yeah, well, it's our pleasure. And as I said in the intro, your lab is focused on the kidney, but why don't we start with you giving us an overview of your lab's general approach for modeling kidney development and what the long-term goals of your work are?
Sure. So the model our lab uses human pluripotent stem cells, and we recapitulate how kidney normally forms in the body, in which actually kidneys is very complicated. But during kidney development, it is actually the self-assembly of just a few types of kidney progenitor cells. So our approach is to recapitulate this process and to build first the building block kidney progenitor cells, and then make use of the kidney progenitor cells to further build a kidney tissue or organ towards regenerative medicine and for disease model in the petri dish.
Yes, I mean, your work has been really critically important, in particular, your work on nephron progenitor cells. It's provided some pretty important insights into like how the kidney can actually build its filtration units during development. And this is such a hot topic for us here on the podcast. We're always talking about innovative new models for studying development, like, these embryo models, which seem to be all the rage these days. But a lot of times we also talk about, the technologies that are enabling the discoveries. So for example, single cell genomics, spatial transcriptomics. So talk about, the technologies, right? Such as these omics technologies that are kind of reshaping our understanding of nephron patterning and maturation. So how have these really taken your work to the next level?
Yeah, so that's a good question. So um, basically, the unique technologies we have in the lab is to be able to expand the nephron progenitor cells. So we first did it with primary cells, isolated primary nephron progenitor cells, isolated from the embryo of the mice, and we developed a culture condition that can mimic the natural niche signals that can maintain the self-renew of those progenitor cells in vivo. And by recapitulating that in a culture medium that we designed with growth factor and small molecules, those primary mouse and human nephron progenitor cells can self-renew, can be expanded for very long term in the petri dish. And then we also applied that to human stem cells and use CRISPR-Cas9 to engineer a dual reporter that allows us to isolate the directly differentiated SIX2 and PAX2 double positive nephron progenitor cells. And then by applying the culture medium we developed for primary culture, we were able to modify it further and allowed us to expand the human stem cell derived nephron progenitor cells. And then we just mimic kidney development cues that's already established to give them a Wnt activation, which is the signal that can activate those progenitor cells to differentiate into nephrons. And then we found amazing self-patterning ability of those nephron progenitor cells to generate different segments of the nephron. And of course, we take different omics approaches to characterize those nephron organoids and compare that with existing organoids in the field. And we found that actually those nephron progenitor cell derived kidney organoids are more mature in a cell type called podocytes, which mediate the filtration function of the kidney.
Hmm.
Yeah, and then something interesting is that we found, kidney differentiation protocols have been there for maybe 10 years. From 2014, 2015, there's already protocol that can generate nephron organoids. So the difference is that we found that if we isolate those transient human stem cell derived nephron progenitor cells and culture those in our medium, they are programmed to a state that's more closer to the primary cells, because we provide additional niche signals to further program them and make them self-renew in vitro. And we believe because of that, when they spontaneously differentiate into nephron structures, those nephron structures are more mature than existing kidney organoids.
Yeah, this is when you lay it out like there, so stepwise, it's so logical. And it seems like you had the plan all hatched, and you just had to go from A to what was that, T? But really, it is so much more complex, and the culmination is so much effort and work. And it is interesting how the observation feeds back on directing the differentiation, right? And now with the single cell, you can see with such high resolution to resolve the niche cues and to resolve what the cells will be responsive to, and the whole spectrum of cells, right? To catch the ones with whatever degree of potency. So it is fascinating to me that although the ideas can be percolating for many years, the technology really props it up, right? So the advances and insight can continue in your hands, getting closer to the real thing, the bona fides, right? A mature nephron progenitor and the derivatives thereof. And we talk about this all the time. I mean, what you just described there is kind of the playbook that has been established with not as much sophistication as you laid it out, but since the early days, right? Figuring out the recipes that now we talk about it every episode are awed by the raft of clinical trials, first in human trials that are coming out, these pioneer works by, like in Parkinson's, for example, from Sierra and Novo Nordisk and Blue Rock, and then diabetes with Vertex and Sana. We just talked about this New England Journal of Medicine paper last episode, where Sana and Sonia Schreppers showed retention of these beta islet cells in human patients. This episode, we just talked right before the interview here about the new paper that they just had showing the CAR T hypoimmune cells, right? And so we were just talking about how hypoimmune really it's not the only future, but it's going to be a major part potentially of the translational application of these cellular derivatives, the actual clinical products that we've all been working toward. But in those cases, like CAR T, in the case of beta islets, there's like this modular function, right? These little units that you can put in. It doesn't have to be a pancreas and the blood. Obviously, it's works on the level of single cells. But I wonder about the kidney, right? My simple mind is just subbing like glomeruli as the correlate of beta islets, right? But is that kind of approach to the deconstructed, the organ analog or organ units, functional units, in this case glomeruli, is that would that work? Or do you need more of the macrostructure and auxiliary cell contribution if you were going to try and reach some kind of therapeutic endpoint with kidney? What are your thoughts on how we're going to get this into people?
Yeah, so that's a great question. That's something I would like to explain a little bit more. So basically, the kidney is different from beta cell or HSC that can work as single cells, right? So it has to be structurally organized. And take the nephron, for example. It has a round glomerulars in which the blood would start to infiltrate. and get filtered in the glomerulus. And then it passes through a very convoluted tubules that has proximal tubule, loop of any distal tubule. So if we do single cell sequencing, we know that just the nephron itself, we have more than 30 different cell types. And they have to be positioned at the discrete locations to perform diverse functions. How can we achieve that and give back the kidney functions to the patients, right? So that's very complicated. So that's why at the beginning, as I mentioned, the kidney, even if the adult kidney is so complicated in their anatomy and structure, developmentally speaking, they are pretty, simple, or they use some kind of mutual induction of a few types of kidney progenitor cells. They can self-organize again and again just by recapitulating the same principles. So as I mentioned, we have developed the nephron progenitor cells. So that's just one building block. So recently, I think we also developed a second progenitor expansion culture and direct differentiation protocols. Now, from human stem cells, we can generate also the ureteric progenitor cells, the UPCs, which can form the connecting duct system of the kidney, and which is actually the organizer for nephron induction and patterning naturally. So amazing thing in our recently accepted manuscript, actually we mix those two progenitor cells together, and we put the UPCs in the center, surrounded by the NPCs to recapitulate the natural niche formation between those two progenitor cells. And what we found was the UPCs in the center would branch out into the surrounding NPCs. And they would also provide a Wnt signaling center, as they would do in vivo, to help pattern the nephron progenitor cells to transition through mesenchymal to epithelium induction transition to form renal vesicles. And though importantly, because the Wnt signaling is in the middle, so it has a gradient of Wnt. So the renal vesicles are actually polarized. It's very different from conventional kidney organoids that have a, not polarized renal vesicle. So those polarized renal vesicle would have proximal end and a distal end. Proximal end will become glomeruli, and distal end will become the tubules that eventually fuse with the connecting duct that comes from the central UPCs. So we get very beautiful structure. Eventually, you will see the publication very soon. So we have like 20 human nephron structures with beautiful round glomerulus and a thin tail tubule, all feeding into the central connecting duct. Yeah, so that's the approach that we are taking. We believe just using single cells of NPCs, we cannot help the patients replace kidney functions. We need to recapitulate the complicated structures of the kidney that has both the nephrons to produce the urine and have the connecting duct that can connect the urine and further process the urine and pass the urine to the bladder to excrete the urine. So that's the proof concept milestone study to demonstrate the progenitor cells do have the potential to self-organize into kidney-like structures. So more importantly, it is not only about the structure in our accepted paper. So in that paper, we also transplanted those organoids. We call it a kidney progenitor assembloid. We transplanted those into immunodeficient mice and get a lot of extensive vasculature formation. So that is key for kidney function, because kidney is to perfuse the blood and generate the urine. In vitro, we cannot do that. But if we transplant in vivo, they can get connected to the vasculature of the host. And they can also perform all kinds of functions. For example, we use the intravital imaging. After transplantation, we inject lucifer yellow through the vein of the mice. After 5 seconds, those dye would enter the glomerular capillary. And after another 5 seconds, it enters the Bowman space and then pass further into the nearby tubules. Indeed, we even connected the fluid and found that it's highly enriched in creatinine and urea, which are the urine components. So this is quite exciting, uh, as a proof concept that if we make use of the combination of progenitor cells and mimic the in vivo development process, we might be able to generate kidney functions upon transplantation. So that's the foundation for our approach. So now we finally have a proof concept. So because of this exciting progress, actually 3 years ago, we got NIH Director's Award. And thanks to the reviewers, we got a perfect score, NIH Director's Award. That allows us to make use of those different progenitor cells to use actually bioengineering approach to assemble the progenitor cells into scaled up structures for transplantation. So that's what we are working on. And we are working on that with a group of kidney developmental biologists, including Andy McMahon, who was our former director of the stem cell department. Now he's at Caltech. And also with a critical expert in bioengineering, Dr. Xiaoshen Chen, who is at UC San Diego with expertise in 3D printing and biomaterials. And we also have a large number of kidney physiologists who help us characterize the kidney prototype we generate, whether they are really performing functions and really can help the patients. We also have nephrologists at USC who help us translate the work in the future into patients.
Wow. We're going to come back around to the dream team at USC near the end, but that is a tour de force, partner. You're killing it over there. And I think it's a really good illustration of how you have to incorporate the native intrinsic, developmental biology and the extrinsic engineering approach, which clearly, the most of the work is the self-organization, I suppose, but it's not enough, right? And that's not to mention the vascularization, but you're hitting it on all fronts. And ultimately, the goal there is to, because it is tough. My simple mind, I'm thinking like, beta islets, glomeruli, but the reality is it's got to be fundamentally, it's got to be integrated, right? Collecting ducts and urine and the blood, everything. It's the incorporation into the system that makes the kidney function, and that's what makes a lot of other organs so tough to reach this therapeutic horizon that we're all gunning for, right? Like the liver. I mean, Arun will tell you the heart. He gets a little depressed when he thinks about, I'm sure, then he thinks about the extent to which you're going to have to engineer something to recreate the complexity of that super muscle, but the kidney too, as you just described. And for that reason, a lot of people have been talking about these genetically modified pigs, right? Xenotransplantation. We talk about it in almost every episode as well, and they're being applied in these increasingly high stakes human trials. And I know this is a bit outside your wheelhouse, even though you've got that whole dream team and the nephrologist, but like the overlap is clear. Do you have thoughts on that approach, or how the engineering approaches of cell-based therapies, creating the kidney and xenotransplantation? Like, yeah, they're complementary. Maybe there's room for both, but are there fundamental problems with either of those that makes one of them closer to reality than the other, or do they complement each other? What are your thoughts on those two varying approaches?
Yeah, so I'm very excited when I see the news that those gene-modified pig kidneys are already being transplanted into patients, and in some cases help patients for a few months with kidney functions. So that's very exciting news for me. And clearly, if we compare those two approaches, it's very obvious that the stem cell-based approach certainly will have a long way to develop. I mean, imagine in the next 5 to 10 years, we probably can get a prototype that can be developed and performing preclinical trials in animals. But clearly, the xenotransplants are already in clinical trials already on the patients. So that's very, very exciting. And we just don't know right now what would be the long-term effect of those xenotransplants. Yeah, so still, this do not prevent us from developing the all human cell-based approach from stem cells.
Hmm.
So I was also, there's also potentially some, combination of those approaches. You know, perhaps we can make some hybrid pig human chimera kidneys that can reduce some immune response if it turns out that in the long run, if all pig cells would not work in the long term. So for example, the nephron progenitor cells that we are generating, it could form some kind of interspecies chimera that can generate all human nephrons, but connecting that stroma from the pig. And also it could have reduced immunogenicity and eventually help the patients.
Yeah, that's an interesting approach. Didn't think about perhaps combining the two, right? I mean, in the cardiac field, it's the same thing. Um, of course, Chuck Murray is the new director of the USC stem cell program, and he knows about this very well, and he's pioneering some of these cell therapies for, stem cell derived cardiomyocytes into the heart after an MI and that sort of thing, and looking at arrhythmias and all that. But why not just replace the whole thing, which is kind of what xenotransplantation is getting at. And I'm not saying one approach is better than the other, but I like your answer there. Perhaps these approaches can be combined ultimately to alleviate the big picture problem, which in your situation, of course, kidney disease. This is huge global health burden, and you need multiple avenues to address that, right? I mean, kidney disease is, of course, driven by chronic injuries that cause scarring, fibrosis, and I mean, are you guys also focusing on the fibrosis side of things? And like, what do you think your role in regenerative medicine might be for treating earlier stage diseases, like before dialysis is needed, before you actually need the transplant? So talk about those kind of earlier approaches that you might be looking at.
I think that's an excellent question. And that is related to the other major research direction of my lab, which is, in the short term, what can we do with those better and better kidney models that we can produce in the lab? So the building an engineered kidney is a long-term goal. But what can we do in the short term to help the patients? We are actively thinking about that and making use of the organoid models that we build for disease modeling. So currently, we are focusing on one of the disease that is called polycystic kidney disease. So polycystic kidney disease, PKD, is actually the most prevalent inherited kidney disease. The prevalence is like 1 in 500, 1 in 1,000. So we introduced PKD mutations into the nephron organoids, and we found that they can form cystic phenotypes, just recapitulate the disease. And we also performed a lot of molecular calculations and found that they can recapitulate the disease hallmarks in vitro. And importantly, we can also, because the organoids are growing in a petri dish, we can develop scalable ways to make thousands of those organoids from each well of a 6-well aggregate well plate. So that allowed us to perform a proof of concept drug screening and identify a potential drug candidate. Yeah, so the most exciting thing is that recently we have moved those in vitro models to in vivo ones. As I just described, we have mixed the 2 types of progenitor cells to generate a kidney assembloid, which is more complicated, more mature, and functional in vivo. So we imagine that if we use those more physiologically relevant model to grow human tissues in the animals, then if we model disease in this type of model, it would be much more high fidelity compared to other existing in vitro models. So we did that, and we transplanted PKD mutant assembloids into live mice. And we found that after 2 months, we transplanted actually under the renal capsule. And after 2 months, the transplant had grown as big as the host mouse kidney.
Hmm.
And if we dissect it out, we were so amazed. It's all full of cysts. Yeah. And then we did a lot of single cell sequencing and compare with patient-derived patient samples. So it is clearly recapitulating majority, if not all, of the PKD features that's already well established, including some of the signaling pathways like cyclic AMP, PI3 kinase, TGF-beta. Those are aberrantly modified in the PKD knockout as compared to the wild type. By the way, wild type, there's no cyst at all, very clean. Okay, and importantly, using cell chat, actually we can see a very clear TGF-beta signaling sending from the cyst lining cells to the surrounding stroma cells. And the stroma cells actually became myofibroblast. So that's a key feature that cannot be recapitulated in vitro. But for the first time in our new study, we now know that this can be recapitulated. And so that answered Arun's question of fibrosis. So I think previously, CKD is chronic kidney disease is featured by fibrosis to mediate the progression of the disease. But that cannot be recapitulated. Now we feel that if we transplant human organoid models, which is already complicated enough, we can generate that fibrosis phenotype and allow us to study PKD, and we can also extend that to CKD so that we can develop early prevention intervention approaches based on those humanized mouse models to study kidney disease.
Wow, so much. You're doing so much, and it's so exciting and you're so understated. I mean, you left that just hanging there. We're talking about building up a big kidney, but the exciting counterpoint there is the modeling, and that can work on I don't want to say micro scale, it's macro, as you just said, your kidney rivaling the size of the host, although it's polycystic, but still. But point being is that you're recapitulating not just the developmental but disease process. That's so cool and so powerful. And it's all sprung from your brilliant imagination for sure. But I think a lot of it had to do with what you got going there at the Keck School of Medicine at USC. I mean, specifically at the Eli and Edith Broad Center, where they've made a huge investment in stem cell biology and specifically like translational therapies, getting young innovators like yourself in early days. And look at what you've done, Zhongwei. You really come through. And you're not the only one. Faculty members among your ilk, regular guests on this podcast. Once every few episodes, we'll have a USC investigator on here, and it's a real nice balance, in my view, of both the veteran leadership with some foundational contributions to the field, as well as the intrepid younger innovators like yourself, who are leading the way to the future. And exemplifying that latter group, you and Justin Achita, another great friend of the show and my own, received this NIH Directors Award that you alluded to just a bit ago, which was meant to support this high risk, high reward research. And you just told us how you're deploying it to scale up the engineering, and with all the support of the nephrologists and the developmental biologists, the whole team there. Sounds like a playground, not just for the imagination, but for your ambition. Tell us about the Keck, why you love working there, and how you think, why and how you think the center has emerged as such a titan in the field. What is it? What's their special sauce there?
Right. So I was recruited to USC at the end of 2017, actually by Dr. Kenneth Harlows who was the chief of nephrology, division of nephrology and hypertension, and also by Dr. Andy McMahon, who was the former director of the USC STEMCELL and chair of Department of stem cell biology and regenerative medicine. So I benefited a lot since I was recruited to USC because of my joint appointment. So I had a basic appointment in stem cell and a clinical department affiliation, working with a lot of nephrologists. So that gives me a unique position to do this translational work, aiming at you know translate whatever we have developed for disease modeling for kidney regeneration to the clinical side. So as you know, Andy McMahon is the I think probably in my mind number one in kidney development studies who has laid the basis for the kidney progenitor biology, right? So, and what I do is to make use of those knowledge and develop new stem cell tools to make those stem cells, progenitor cells, and try to assemble them together. So I benefit a lot by discussing with Andy McMahon and to learn some of the most updated information about the kidney progenitor biology, so that can guide me through the differentiation protocols and expansion protocols. Yeah, there's a lot of collaboration, and even after recently moved his lab to Caltech, we are still collaborating a lot. And also another kidney development biologist, Niels Lindström, we also have a lot of collaboration at USC. In addition, there's a lot of excellent kidney physiologists who can help us characterize the kidney organoids in terms of their functions. As I mentioned earlier, Dr. Janos Peterdy, who is a director of the intravital imaging core facility at USC and a very famous kidney physiologist, who is just across the bridge of our building to the neighboring building, and he can provide intravital imaging to help us, evaluate in real time the kidney functions achieved upon transplantation. And there are also other kidney physiologists. Susan Gurley is our new department chair of medicine, and also Alicia McDonough, and at CHOA, Laura Perrin. So many excellent kidney researchers at USC. And I would like also to mention Dr. Angie Chang, who is at the Viterbi School of Engineering, who is excellent in making nanoparticles to mediate any drugs into the kidney specifically. Mm-hmm. And she was also awarded NIH Director's Award for that very cutting edge nanomedicine research. So we all work together and to push forward kidney research at USC. So that's for the research part. I also want to mention that at USC, there's a very unique foundation called UKRO, University Kidney Research Organization. So it was founded, I think, back in 2002 by a grateful patient who got kidney failure and got a kidney transplant from the former chief of our division of nephrology. So they founded this foundation to support kidney research at USC, and they are providing a lot of fundraising activities to raise funds to support my lab's research. So that's really, really helpful, as you know, especially in this climate, right, where the funding is extremely difficult to get. So yeah, so that's another benefit at USC.
Yeah, sounds like LA is the place for kidney, and USC in particular. I know you've got some friends nearby at Cedars-Sinai as well. Dr. Sanjeev Kumar, for example, who you've worked with. So quite exciting to share and talk about the work you've been doing over the years, Zhongwei, and we're excited to see that paper come out pretty soon. And before we let you go, we're going to ask you some peripheral questions just so our listeners can get to know you a little bit better. So first of all, if you could answer like any scientific question, and doesn't have to be in stem cell biology, regardless of your expertise or regardless of your chosen field, what would that question be that you would like to tackle?
Yeah, I think one of the challenges I think in the biomedical research field is to understand aging. I think aging concerns everyone, right? Especially I think at my age, every time I came back to visit my parents in the summer, I know that I just can notice that they aged year by year. That's something I definitely want to stop. if I would have the magic power, I would like to know how to stop that and help people slow the aging or get very healthy aging.
I mean, as I have a special guest nearby with me on this podcast, as my son, who's like kind of the opposite of old right now, is joining us, can attest to, yeah, aging is something that we're all going through, and I think that's definitely a very relevant question to answer. And then finally, the last question we'll ask you before we let you go: what's the best piece of advice that you've ever been given, professional or not?
Right. So I want to share, the advice that I got from my postdoc advisor, Dr. Juan Carlos Ispishua Belmonte. So before I left Salk Institute and moved to USC, we had a farewell party, and I asked Juan Carlos, "What would be your advice for me to become an independent PI? Would the life be easier? And what do you want to say to me?" And he said, "Your your work is gonna be even more and more challenging and more busy. So, but remember, um, grant will come, papers will come, and the most important thing looking forward for you is that you need to make sure that you enjoy every day with your lab members and enjoy science. That's a key." Okay, so I think I always remember that, and as he predicted, papers came, grants came, and I really enjoyed every day with my lab members. So that's very important piece of advice that I have received.
Yes, I mean that is all those three things weave together in success. But yeah, it just starts with that, right? The joy of observation and sharing it, right? So words to live by, a great piece of advice for any young trainee. Don't ever forget the joy of looking down the microscope or whatever you're looking at, making that first observation, because that's what you'll take with you forever. And I'm going to take a few things away from this interview forever, I'm afraid, because you've really blown my mind, Zhongwei. And I appreciate you being so forthright and sharing your unpublished work and balanced in your, review of both the big macro engineering problems as well as the micro modeling molecular biology. You're really firing on all cylinders, and it's been a great joy hearing you share with us and our audience. So thank you so much for joining us.
Thank you so much. I really enjoyed it.
All right, guys, that brings us to the end of this episode number 302. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com, where you can get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCellPodcast.com, or via email at info@STEMCellPodcast.com with feedback or to suggest guests. Summer's almost over, guys. So you've got to get back into the hood. Good news is we got episodes coming every other week.